Speaking. No, somebody has my thing of tweezers. Not I. I don't know where they are.
Okay.
False. Okay. Let's take a look here then. We're going to call this lesson 3.7 Gene Expression. I almost did the less common spelling of lesson there with an M at the end, but we got this. It's a variation. That's right. Hello. Do you need a sharpener?
No.
Okay, cool. All right, so we want to talk about gene expression. So we have in the human genome about 20,000, give or take, unique genes that code for polypeptides. And that 20 to 80,000, how is it that 20,000 genes can code for 80,000 polypeptide sequence? Different splicing patterns. Does that make sense? What body is responsible, or what complex is responsible for splicing? Oh, the spliceosome, indeed. And what are the different segments of DNA that are typically included called? Exons. Good. So we can keep different combinations of exons, which gives us those. Now, these undergo further modification after translation, i.e. once they are polypeptides. Which gives us more unique proteins. And there's about roughly 1 million unique proteins in the human, what we call the proteome. So I just want to draw that. You don't necessarily have to draw this if you think you get it, but the basic idea is we have our DNA, right? And we could think of that as like single strand or whatever, but we have the genome, all right? And the genome is the collection of all our genes, and we have roughly, very roughly, about 20,000 coding genes. Different scientists will count different segments as individual genes versus kind of breaking into sections and things like that. So there's not a perfect exact number, even though the human genome has been completely mapped. Deciding where one gene starts and another ends is sometimes really obvious, and other times it's not. By what process do those genes get turned into mRNA sequences? Transcription. So by the process of transcription, we can get an RNA strand. And of course, that RNA strand might have different segments that were desirable and different ones that weren't, such that a single strand of RNA can produce potentially, oh, I don't know, 4, 5, 6, 7 different things there. So we can have splicing and modifications. Yeah, what's up?
I just wanted to let you know that I found your 4 steps. Me, I don't have them, and Trash has them right now.
Ah, well, can you make sure they come back at the end of the class, because I've got a bunch here that I need to join to them? Thank you. And we call this the transcriptome, which is a stupid word that you'll never actually use. But so the genome refers to the collection of all your genes. The transcriptome refers to all of the mRNA sequences that can be made from that. So these are all mRNA that will then go. And where does the mRNA go? Out into the cytoplasm, where it undergoes what process? Translation. Good. And when it undergoes translation, what do we get? We get a big polypeptide, right? So here it is. That is a polypeptide. And we have roughly 80,000 possible polypeptides. And from there,
What? I don't got room for this stuff.
sorry, I won't tell you. Try and narrow down. You're creative. If you found that you really went too big, you could just highlight it, kind of like this. Let's give it a little highlight like that, and then what you can do, there's a little corner. If you grab that and you drag it like this, it'll shrink a little, and that fits a bit better. You're welcome.
Very helpful, thank you, Mr.
I know, right? And this can fold and be modified and spliced itself in different ways. And maybe we get that same polypeptide can make 3 different proteins. And aren't my proteins beautiful?
Looks like modern art.
They're very good, right?
So we all understand modern art, and we just think it's bullshit.
Okay. So different foldings. We get combinations of different ones, right? And we get removal of sections. All of which leaves us with roughly 1 million proteins. Does that make sense? Now, there's nothing we've just drawn that we hadn't written in the words above, but I think sometimes drawing it out is helpful. Yeah.
Just like how we have genome and transcriptome.
Wait for it. Wait for it. You know. It's the human. It's the proteome.
Oh, but the peptides don't get any?
Not that I'm aware of. I think like this kind of collectively would be considered the proteome. And we refer to each of these as an individual protein species. So different species of the same polypeptide. So we could call these species of proteins. Sorry? Okay, so we got 1 million proteins. Awesome. Does every cell use every one of those proteins all the time? Of course not, right? Does that make sense? Like not even remotely. Some of these proteins get used almost constantly by the cell, and some of them will never be used by any of your cells. We have some proteins that our DNA codes for that simply are not used, to the best of our understanding. But not all of these are used equally. And I'll remind you that you should never start a sentence with a conjunction, but here we are. And I don't care. And different cells have different functions. So the question, of course, is how? How is this controlled? So I'll give you a moment to write that, and then we'll talk about how it's controlled.
Like what? Like how the cell knows what?
Yeah, sure, right? Like you've got, how does your cell know, hey, this gene here, we better make a copy of that right now because we need that protein. But let's not make a copy of this section here, no mRNA from there, no mRNA from here, right? Like how does your cell know which regions of the DNA to copy and which ones to leave alone?
Can our cells really know something?
Mm.
Is it haplogroup?
Well, that is that thing we all do where we use sort of anthropomorphic language to describe things that absolutely aren't human, right? So you're absolutely right. The cell isn't making decisions. It's not like the cell council gets together and has a meeting and reviews the annual protein production, right? It's not like that at all. So what is it like? How does it work? Well, we're going to talk about transcription factors. This is one of the pieces of the puzzle, right? Or TF is, that's not like some official short form. I'm just putting that there because boy is that what I'm going to write every time I need to write that word in the note ahead. Does that make sense? So just to pre-warn you. My desire to write out the word transcription factor 15 times is limited. I mean, not 15, but... Okay. So those regions of the DNA ahead of genes and when I say ahead of, do you know what I mean? Like sort of before, if we're reading in the 5' to 3' direction, before the section is the gene, that is the gene, there's a section of the DNA that we call a transcription factor, and it's designed, I shouldn't say designed is that has a high affinity, that has a high binding affinity to specific proteins or RNA factors sometimes. Remember how we talked about how sometimes RNA can work as an enzyme? Sometimes transcription factors will bind to little segments of RNA. So we'll say to proteins or sometimes RNA factors. Sometimes the binding protein encourages transcription. i.e., it would attract what? What is the enzyme responsible for transcription? Very good. So it would attract RNA polymerase. So we have these regions of the DNA that have a high binding affinity to some specific protein or some specific RNA factor. And we'll talk about what that looks like in a bit. But sometimes the result is that once that binding protein has attached, that encourages transcription. We talked about this before, and if you remember, we call these activators. Or sometimes we'll use the term positive transcription factors. Positive transcription factors. So sometimes the binding protein discourages. And again, obviously, discourage and encourage are very anthropomorphized terms, but discourages transcription, i.e., it repels. RNA polymerase. And we call those we've used the term. Very good. Inhibitors. Boy, when you write that word slowly, it begins to look ridiculous. But that is, in fact, spelled correctly. Sometimes we'll call them inhibitors. Sometimes we'll use the word repressors. Or sometimes we'll use, and you should be able to predict the third one based on what we wrote above, negative transcription factors. Very good. Good. Okay, and I'm going to use, have any of you taken physics? When you took physics, did everyone see that? That got crazy. It's a very nice looking waterfall. Okay, whoa, wow, that got big. Okay. Oh yeah, the picture is what I wanted. Thank you guys. That's what I wanted. Okay, so we're gonna go here to this PhET simulator, and I want to take a look here and see this bit on gene expression. Does that make sense? So let's take a look, and this actually shows the basic idea, where we have a gene here, and this is what we call the transcribed region, i.e. this is the portion of the gene that is going to actually get transcribed, right? And this is what we call the regulatory region. Does that make sense? Mm-hmm. And so if I take RNA polymerase and let it float around, if I put it right on here, look what it does. Right now it doesn't bind because it needs to be kind of encouraged to do so. So what else do we have?
Use your transcription factor.
I have what's called a positive transcription factor. And if we just let it float around, it will eventually find its way there. To speed things up, I'm going to put it there. But it would actually find its way there sort of by default. And to speed things up, I'm going to put that there now too. And we can see, isn't that fun? That's fun, right? But that was fun, right? And what do we have now?
That's just a...
What is that?
It's like a protein. Lots of more protein.
Is that a protein?
Enzyme?
Does RNA polymerase make protein? What does RNA polymerase make? mRNA. So that is a piece of mRNA, which would need to make its way to the ribosome. Does that make sense?
Lots of mRNA.
And so what did it just make? A protein. Isn't that fun? But now look, that piece of mRNA.
You don't have any proteins yet.
That's a protein right there. Should I capture it? Look at me. I've got a protein.
Okay, thank you.
And the other thing we have is that these will just kind of float around, and again, if it finds its way to another ribosome, it'll make another protein, right? And if it finds it again, it will make another protein. And I am, when we see later simulations, see, you don't have to make it. They will eventually bump into each other. Just like these will eventually bump into here, but of course, if I don't want to wait, I can just put it there, right? Now, of course, the other thing is, do we want that mRNA to be working forever?
No.
No, what? Do I need 1, 2, 3, 4, 5 of these?
No.
And will it just keep getting made? So what else we have is mRNA destroyers, right? And they can float around, and if they happen upon the mRNA, they will
eat it.
Or they'll eat it real good. Does that make sense? All right.
Why is that ribosome on the DNA?
Because things float around, and it is an important thing to understand That is actually a big part of how all this works, is that we talk about things going here and wanting to go there and do this. The fact of the matter is things float around, and when they bump into something for which they have an affinity, they will act. So if this were over here, it likely will make its way down there, but there's no guarantee. But it has a high affinity, and so, oh, see, it's oh, look at that. Look at that. Didn't make it. Looks like it made it. Didn't make it. Right? Okay, so let's look at that in a bit more detail. We'll put all the various pieces out here. So this is just another example of a region that has not one but two transcription factors. And with those present, it can do its thing. Remember we talked about how you could actually have RNA polymerase acting twice or even 3 times on the same thing, right? Get over here. Get over here, you. See, and they do make their own way, and these will make their own way, and left to their own devices. But of course, you can also have a negative transcription factor.
We've made more proteins. We've made hexagonal protein. Wait, how many sides is that? 1, 2, 3, 4, 5, 6. That's right, hexagonal.
I'm struggling to get my negative transcription factor to attach.
The positive transcription factor keeps trying to get onto it.
Anyway,
Can you capture them?
but you can see we are now making.
Can you put those proteins in the collection?
Do you want me to put them in the box?
Thank you.
Okay. Is that better?
Yeah.
Okay, now the next one we have to capture 3.
Yeah.
Okay, I like that. Okay, so here, again, we have 2 positive transcription factors, 2 of these. And if we were patient types, we could just wait for everything to make its way where it needs to be made, but I'm not patient, so I'm going to do this. And that. Oh no, it really does feel like the Pac-Men are going to... Oh, look at that! Oh no! Oh no, it didn't even get to make it. It didn't even... Oh, that little bastard. Look at him.
Oh, what is
Oh,
that?
he left that.
He's vicious. He's vicious. What a mean protein destroyer. Or mRNA destroyer, rather. You get over here.
The hexagons are coming back.
Oh, wow, the hexagons are coming back. There we go. Okay, these guys need a little help. Get over here. Get over here.
There's another hexagon.
No.
Where are these hexagons coming from?
From the last side. Because if you looked, it's all one screen, actually, just moving around, right? Like, so we didn't turn this off, so they're still going. They're still doing their thing.
There's another one.
The squares didn't do anything.
See, whereas over here, things are still getting made, but they're not floating their way over.
Oh, I have proteins. Can you collect it?
But you're actually seeing a bit of the problem, which is, what am I still getting a ton of from the previous page? My little hexagon proteins, right? Which maybe I don't want 8 hexagon proteins. Maybe like I want them to like chill out, but they're not, right? So the basics of gene expression are that if I went back to the previous slide and removed the positive transcription factor, right, we would actually stop getting all these hexagon proteins, at least in theory. Though, boy, that negative transcription factor really is struggling to get a good binding.
It doesn't help that the mRNA destroyers are just kind of...
No, but they'll get to it eventually.
They were relentless 2 seconds ago.
No, right? They're vicious.
They don't seem to care much about the...
Well, this guy's just really attracted to the last screen button. I don't know if you saw that, but it was like, all right, last screen. Whereas if we go over here, we're going to be getting like green stupid things like crazy, right? And they still are, because the positive transcription factor's on there, right? So that's the basis of gene expression, which is that if we remove kind of the promoter segment, it doesn't mean that transcription will never happen, right? Like it did happen here, but it slows it right down. Does that make sense? If we went back to the first one and remove the positive transcription factor and instead place the negative transcription factor, this gene won't express at all, and we will stop getting our little diamond, or square if you like.
That it worked out in the end. That's a nice version.
Okay.
Nice
How's that feel? Reasonably okay? Okay. From there, I want to give a specific example.
Gene machine.
The gene machine.
Sounds like a TVO kids show.
That actually does sound like the
It does, doesn't it?
Jar is loaded.
Okay. So I want to look at a specific example here, which we'll come back to. So,
I feel like I'm being bullied.
sorry?
Lactose. I'm being bullied right now.
He's lactose intolerant.
I'm very lactose intolerant, but I still put myself through it. I'm not, what's it called?
Well,
Evolved enough.
You're not what?
Evolved enough.
You're insufficiently evolved?
Yes.
Well, maybe lack intolerance is the next evolutionary step, right? Who knows?
People are actually born intolerant to lactose, but some people are more tolerant to it.
All right. So, example, the lac operon. We're going to use a bacterial example because bacteria is simpler. Does that make sense? So, bacteria will always use glucose as a primary food source, will always use glucose as an energy source if available. But if no glucose is present, bacteria will use lactose if available. I'll say for energy. And lactose is a disaccharide. What's that mean? Yeah, and it's a disaccharide of galactose and glucose.
Is it supposed to say bacterial lactose?
Sorry, what's that?
Oh, sorry. I was just seeing things.
Okay. Try to stop interrupting my class. Good Lord.
Sam, we're really disappointed in you.
What are you not though?
Yeah, it's true. I'm glad you understand that.
Those look like some strange glasses.
I need tilling acid.
So that's lactose. And there's an enzyme, and the enzyme is called beta galactosidase.
Wow.
Beta galactosidase. Right, so that's an enzyme. And of course, an enzyme is a protein. So that's a protein. And it will break this up into galactose and into glucose that the cell can then enter into its metabolic pathway. Of course, what is that metabolic pathway? Yeah, cellular respiration. What's the first step of that? Glycolysis. So these can then enter the glycolytic pathway. Does that make sense? You did kind of whisper. I'm an old man. I'm deaf. You gotta talk loud if you want the credit. That's just how it goes. I don't make the rules. Um, beta-galactosidase do we need to write this? It's there. There's no point writing another sentence there. Rather, we'll say this. There's no need for the cell to do this if concentrations of glucose are high. And are people comfortable with the square brackets around something meaning the concentration of? If you took chemistry with me, you'd be used to that. If you didn't, just a note that square brackets means concentration. In other words, if we have lots of glucose, why would we bother wasting time and energy using a protein to break lactose down to make glucose? Does that make sense? It would be like if you open the fridge and there's a bunch of like, oh God, this analogy's getting away from me. If you went into the kitchen and you're like, your mom or dad had already like cut up a bunch of like fruit and vegetables, and you're like, I want delicious fruit and vegetables cut up. You're not going to go into the fridge and get out a bunch of raw vegetables and peel them and clean them and wash them and cut them up. You're just going to use the ones that are sitting out, like that have been nice and freshly prepared, right? Does that make sense? If you're like, boy, I could really go for chicken fingers right now, you're not going to go get your crossbow and go hunt for a chicken, right? You're gonna like get the ones out of the freezer and put them in the oven, right? And then forget about them and come down in the morning and the black charred. You ever done that?
You've done that. That's
Oh, I've done that. Oh, you know I've done that. So, when should the cell make beta-galactosidase? If beta-galactosidase is the enzyme that allows a cell to metabolize lactose, when should the cell make it? And I thought we could make a little table. That make sense? I thought, you know what people like? And you got to give the people what they want. People like a table, right? You know what people really like? If you're ever in a job where you have to give reports, God, do people love a pie chart and like a bar graph. It is amazing how if you've got like, you got information you got to tell your boss, and you go to the trouble to make like a pie chart or a bar graph, which seems like grade 6 stuff. I'll tell you right now, they're always like, oh, that's a good report, Orlando. I'll be like, oh, no problem, you're welcome. People are always very impressed by pie charts. People are like, I can really see that now. Anyway, we're not going to make a pie chart here. We're going to make a table. But I'm telling you, I can remember months when I'd had like really bad sales reports and things like that, but made a really nice looking report. And at the end of the meeting, my management, like the owners would be like, good job. And you'd be like, no, I objectively did a bad job. We had a terrible month. But you're right. It's a good looking report. Anyway. All right. So here's the basic idea. What? One more time?
pretty privileged, but it's reports.
You'll be shocked to hear that I'm not well acquainted with the idea of pretty privilege.
Pretty privilege.
Sorry?
Pretty privilege is totally a thing.
Oh, it's absolutely a thing. I assume this. I know this as in math. There's proof. There's direct proof and indirect proof. So I think my life stands as an indirect proof of pretty privilege. Which is all the things I don't get to do by being unpretty. But I feel pretty witty and wise. Okay, so let's look at glucose levels, lactose levels, and let's talk about the transcription rate of the gene. Okay, does that make sense? So, if we have high levels of glucose and no lactose, like low or none, are we going to make this gene? Remember, what does this gene do? This gene breaks down, it makes a protein that breaks down. Yeah, it makes an enzyme that breaks down lactose. If we have low levels of lactose and the cell already has lots of glucose, are we going to transcribe this? None, not at all. Does that make sense? It would not make sense to. What if we have high levels of glucose and there's lots of lactose around? What then?
Not really, no.
Yeah, it'd still be very low. Might not be none, but it would still be very low. The fact that we have lots of lactose means that the cell might want to start breaking that down, but still, its main energy source is going to be glucose. What if we have very low glucose, but we have no lactose available? Do we want to be making beta-galactosidase? Is that a good use of the cell's energy? No, because what does beta-galactosidase break down? Lactose. And if we have no lactose, is there any point in making this? No. So what will be made? None. So the only condition in which we actually want to make this is if we have low levels of glucose and high levels of lactose. In that case, we want to make a lot of beta-galactosidase so that we can use the lactose as an energy source. Does that make sense? This would be, if you were sitting down and designing this system, this is how you would sit down and design it. You'd say, okay, we only want to use lactose when we need to, right? Because it's not the most efficient source. Does that make sense? Is that comfortable? If we were designing it, that's what we would design. As it happens, that's how it evolved. The bacteria has evolved to only metabolize lactose when the only food source available to it is lactose, which is efficient, right? Does that make sense? Okay, so how does this work? Okay, you ready? Are you ready? This is a little complicated. We ready? Okay. This is regulated by 3 genes and a regulatory region. So it's regulated by 3 genes and a regulatory region. So we're going to draw a little diagram. We're going to diagrammatically draw our DNA. And we're going to show our DNA thusly. And I'm going to go back and change that to black, actually. That way I have blue as a color if I need blue as a color. Right? Okay, so this is going to be our strand of DNA. Okay? And there are 3 genes that code for things. And those genes are called LAC-Z, LAC-Y, and LAC-A. Boy, is that a weird naming system. You know who didn't name them?
Whoever made the alphabet?
I was going to say me, so I don't want to hear about it, because people always complain. But yeah, it's brutal. I don't know, like, oh yeah, Z, Y, and A in that particular order. I don't know what to tell you. I didn't come up with it. I think it's silly, but here we are. Oh, golly, that's a big highlighter. Did I just unironically say, oh, golly?
Yes.
I like it. So these are 3 genes, and 3 genes are going to code for 3 proteins, right? So here are the 3 regions, the LAC-Z gene, the LAC-Y gene, and the LAC-A gene. And there we go. I was going to use purple. I was going to use variations on. Yeah, I like that, I guess. Okay, so these are protein coding genes. And then over here, we have the regulatory region. And the regulatory region has, okay, actually another protein coding gene, right? Another protein coding gene. And over here we had LAC-Z, LAC-Y, and LAC-A. Here, of course, we have LAC-I.
Yep, sure.
This one actually makes a little bit of sense because I stands for inhibitor. All right. LAC-I also codes for protein. In this case, it's the inhibitor protein. And then we have a sequence of regulatory regions here. We have O for operator, CBS, which we'll talk about.
The Canadian Broadcasting
No.
System?
Canadian Broadcast.
P, which you could probably guess what it stands for. And O. Okay. And how did I have those colored? I think I coded sorry about that, the operator is in orange.
Whoa, that is dark.
I'm sorry.
You can't control how your highlighters turn out.
Well, I mean, I kind of can, but anyway. And I used green for the promoter. Okay. Does this look complicated to anyone?
Just a little
Looks
bit.
like a rainbow.
Yeah.
Slightly complicated.
This is just an example of how a single gene is regulated. Yeah, Manat?
Are we still talking about bacteria?
Yeah. We have a similar system. Ours is a bit more complicated. So we start with the bacteria. We don't even start with. In this course, we just end with the bacteria one. This is an example of just one gene. How does the cell control when to make this one enzyme? Again, we have a million different proteins. This is the control mechanism for one of them, which is pretty, I think, in a way, humbling, right? Okay, so what's going on here? Okay, well, we've got these genes, right? What do? So the lactose metabolizing genes are grouped together, and there's 3 of them, and as it turns out, LAC-Z codes for the enzyme. That beta-galactosidase enzyme. LAC-Y codes for a transport protein that will move more lactose into the cell. So the cell is like, well, if we're going to break down lactose, then while we're at it, we better make sure that we keep bringing lactose into the cell. So LAC-Y codes for a transport protein that moves lactose into the cell. LAC-A codes for a protein, and we don't really know what its function is. It's a protein of unknown function. That might have changed since I last studied this and made this note. That's something I might look up. I probably should look that up in advance. But we just mention it as something we include because it does get made. It's part of this same promotion system. Okay, so now what is the transcription factor here? What is all of this, the regulatory region, right? Because all of this is regulatory. And when we say it regulates something, what does it mean to regulate something? Well, it means to say, yeah, we're going to transcribe this gene, or no, we're not. So let's talk about the transcription factor here then. So the inhibitor gene is a small little protein, which means it's much easier to make. It's much less energy for the cell to make this inhibitor protein than it is to make the beta-galactosidase enzyme, which is a big complicated enzyme, right? And this inhibitor gene codes for this inhibitor protein that binds to the operator and stops transcription. But it will be attracted to lactose and leave the operator region, i.e. leave the operator region. Got to give Barry a trip here. Where is Barry? Barry's back there. Listen, you guys can all, we're going to call this the equivalent of Barry. There we go. Okay.
You put it in there.
So it will leave the operator region and go to lactose. So basically, the cell has this small little protein that it can make. And when it makes this small little protein, that small little protein will bind to the operator and it will turn off the gene unless lactose is present. So the result of this is lactose is required to remove the inhibitor. Which means if there's no lactose, no beta-galactosidase gets made, no transport protein to move lactose gets made, and none of that third protein, which we don't know what it does, gets made. Okay. Yes?
So does LAC-A just code for something that we don't know?
That's right. Yeah, we don't know what it does. It does code for a protein. It makes a protein. We don't know what that protein's function is right now. We presume it's probably involved in breaking up lactose, but we don't know its specific role. Okay, now that satisfies half of this, right?
Okay.
Because that satisfies that if lactose levels are low, we would get no transcription, right? But it doesn't get us all the way, because in this situation, that system alone would mean that we start making beta-galactosidase. But actually, in this case, even if lactose levels are high, if glucose levels are also high, the transcription factor doesn't get made. Does that make sense? So there is a second layer of control. Oh, you're welcome. Just when you thought this was too easy.
Is it the CBS?
Sorry?
CBS?
Canadian Broadcast Station.
Yep. CBS stands for the cyclic AMP binding site, if you're wondering.
Can you say that one more time?
So,
AMP go?
well, just one second. Layer of control. Well, so fully it stands for the cyclic adenosine monophosphate binding site, but we don't want to call that. Okay, so we have our negative transcription factor, right? This is a negative transcription factor, right? It inhibits. But there's also a positive transcription factor, all right? There's a region called the cap site. Or sometimes the CBS site before the operator. Of the lac operon. All right? This is a positive transcription factor. And it requiring a molecule called cyclic AMP. Do you want me to write out adenosine monophosphate? Oh, hell, I'm going to do it. Yep.
Is the AMP like related to like ATP?
Yep. Yep. Except instead of having 3 phosphates or 2 phosphates, AMP has 1 phosphate. See how the cell reuses the same materials over and over and over again? Yeah, Manette. Right here, that is the cyclic AMP binding site. So this can also be called sometimes the cap site. We'll get there. They're all involved. You'll see. When we actually animate it, you'll see. The operator there's 2 places for operator binding, and then we know what a promoter does, right? The promoter is where what binds? We thought in the simple animation, the promoter is where RNA polymerase binds, right? So before RNA polymerase can bind, so now how clever is this? Because of how it works, when glucose levels are high, ATP is being made, right? And as it happens, to make ATP, we haven't talked about this before, but cyclic AMP gets turned into adenosine diphosphate, and adenosine diphosphate gets turned into adenosine triphosphate. So, if there's lots of glucose, if there's lots of this, there will be very little of this. So this is the second layer of control, which is that for this gene to get used, it has to bind to something that is only around when the cell is low on energy. Does that make sense? If the cell has lots of energy, then it won't have much of this. On the other hand, if the cell has low amounts of this, well, then it will have lots of this. Does that make sense with my color coding there? I'm showing 2 possible cases, right? So in this blue case, this would be cell is lacking glucose. And the red case would be with lots of glucose available. And this is the regulatory regimen. because what it means is that if lots of glucose is available, we're not going to have much cyclic AMP, which means that it can't bind to the promoter. And if it can't bind to the promoter, it doesn't matter whether there's lots of lactose available or not, because unless glucose levels are low, cyclic AMP levels will be low, which means it can't bind to the promoter. And if it doesn't bind to the promoter, then RNA polymerase can't bind. And if RNA polymerase can't bind, then the gene doesn't get transcribed. Does that make sense? Okay, so let's look at that. If CAMP levels are low, you know what? Just leave me alone. If cyclic AMP levels are low, the CAP site sits empty, and transcription doesn't occur. Regardless of lactose levels. Now, you might be wondering, why does that work? One note, just one thing to note, cyclic AMP doesn't bind directly to the operator. But to a protein called CAP. And that's why we call it the CAP binding site. And if you're wondering, is that the cyclic AMP carrying protein? Yes, it is. The names are brutal, right? This really means that there's three possible scenarios that we'd have to consider here. We'll get through all three of them today, but maybe we will. The first would be if there's low lactose levels. And if lactose levels are low, I'm going to draw a slightly simplified version of our DNA here, where I'm just going to call one section genes. We know there's how many genes actually? There's LAC-Z, LAC-Y, and LAC-A, but I'm just going to call those the genes here. And we can put in brackets LAC-Z, LAC-Y, and LAC-A. And before the genes, we have the operator. And before the operator, we have the promoter. And before the promoter, we have the CAP site. Okay, remember, the promoter is where RNA polymerase is going to want to bind. But RNA polymerase can only bind if there's something present on the operator. All right? And what we're going to find here is that on the operator, we have the inhibitor protein. And there's no lactose to attract it away. And in that case, it doesn't matter if RNA polymerase binds here, all right? We know that RNA polymerase is going to bind at the promoter, right? But if it binds at the promoter and tries to go forward, what's going to happen to it? It's going to be blocked by the inhibitor. So this blocks RNA polymerase. And so even if RNA polymerase binds to the promoter, what doesn't happen? They can just go on the center table back there. Thanks so much, kid. They don't get produced. Does that make sense? So, so there's no transcription because the inhibitor, which is made by the LAC-I gene, is binding to the operator. Does that make sense? Okay. Scenario 2 would be that we have high lactose, but we also have high glucose. And in that case, the operator is going to sit empty. The inhibitor won't bind because the inhibitor will be bound up with lactose. This is my attempt at drawing a lactose. Right, these are our genes here that we want transcribed. We have the promoter, but in this case we also have the cap site, and if the cap site is empty, because there's no cyclic AMP available, then RNA polymerase won't bind to the promoter. All right? So until the cap site is full, RNA polymerase won't bind to the promoter, and so even though the operator is clear, the promoter won't bind, and as a result, the genes don't get transcribed. Okay, and that leaves us to our third situation, which we will cover tomorrow and do the actual simulation. Yeah? What makes that happen? Where does the CMP come from? CMP is always present at some level in the cell because it's a precursor to ATP, and the cell always has ATP present. Now the thing is, because it's a precursor, if ATP levels are high, then levels of all the precursors, the levels of cyclic AMP and the levels of ADP will be low, because these get made into that. Does that make sense? If, on the other hand, the cell has used up all of its or not all of, but used up most of its ATP, and it doesn't have extra ATP available, well then cyclic AMP levels will be very high. So cyclic AMP levels are a way of indirectly measuring ATP levels, and ATP levels are a way of indirectly measuring glucose levels, because if the cell has access to a lot of glucose, it'll be making lots of ATP. Did that answer your question? Do you see how complicated this is? Right? Like, this is like 6 degrees of separation, where it's like, we want to know whether we need to make beta-galactosidase, this enzyme that breaks down lactose. So we need to know, well, is there a lot of glucose available? Because if there's lots of glucose available, we don't need to transcribe this gene. and it doesn't measure the glucose. What it reacts to is actually how much of this stuff is available, and this stuff's availability is related to how much glucose there is. Because if there's lots of glucose available, this stuff's going to be constantly getting made into ATP, so there won't be much of it. But if the cell is starving for glucose, this is going to start to build up because the ATP is going to be getting used up and turned into ADP, and some of that ADP will decompose back down into cyclic AMP. Was that helpful at all? This is complicated stuff. I remember long nights spent studying this in my grade 13 biology class, like trying to be like, oh my God, there's going to be a Laff-Copperon question that's going to kill us. But you'll get it. We got it. And you're smarter than I am, so you'll get it. Yeah. I would love to. So we'll pick this up tomorrow. Good news. This is the last lesson in this unit, team.
Yay.
Huzzah. And then I'm giving you at least a week where we're going to be doing some stuff, but you'll have time to review this stuff and really make sure it sticks in there.
We're still having issues with the bell, so at this time, students can start making the way to their period.
Okay, but there was a bell at 8:05 this morning, right? I swear I heard it. No, I'm sure there was. I was walking in. I know because I was late and I was like, oh my God, the bell just rang. I'm like, well, at least it's only the first one. I gotta go grab my test and staple them. How you doing? How was your weekend? Good.
So I can review questions?
I'm gonna say no, because I gotta go get this. The quiz was good. It was solid. Hi, how we doing? Good.
You're gonna build math this? Sam, I haven't been here. We're talking about math. Oh. You want to review the note page or something?
Okay, you're probably gonna fail that. I'm so glad I
We have math today. He moved it for us today. To be honest. I don't know. I wasn't here.
Why is there a bunch of balloons? I hate this so much. I can't eat solid food till Thursday. I haven't eaten anything other than apples since Monday.
I know anything about the water.
You had?
I think it's me if I only ate applesauce.
I'm gonna be it. I'm already tiny. I lost like 20 pounds over the summer. Where did that weight go? It's not good. I had a nice pair of shorts really like, and then I don't fit me anymore. Just eat a lot of apples. They fit me in July. I wore them on July 6th. I remember it, and they fit. And then I put them on in September, and they.
He forgot his seconds.
He drank that.
So,
You were about to understand. I was out for work. I saw him bring his bag.
And Carmen didn't get me when y'all were sick, so I'm gonna have to wait till volleyball season where she has
What does Kat do? Put a bunch of blood black?
Yeah.
Okay.
Not gonna think
Over
about
the
it.
weekend, you might not wanna have
Black, whatever.
I don't know if I'm sick or if my melatonin is on my nose.
My shoulders hurt so
I
bad.
can't feel them at all, and my hip.
My hip. Okay, this is
I'm
a
so
bounce
fucked.
mark. Or whatever
What is that thing
balls.
where like you're in between people?
Seam?
Oh, see, Orlando, we should teach through volleyball. See.
Okay.
Do we not have tryouts today?
We do.
We do? Didn't it say on the announcements that junior boys basketball or whatever was today?
Oh,
No, I said Wednesday, Friday. Listen better, guys.
Oh.
Okay, to be fair, you were really quiet.
Yeah, you're right. Sorry, just give me a second.
I heard Monday, Wednesday.
Just give me a sec. Okay, guys, sorry. I gotta get these stapled. Shh. Shh. Talk amongst yourselves.
Oh, yay. I can barely raise my arms.
I woke up, I'm like,
Sean put them together. I put them together. Usually my skin is like, but I like just cannot.
we had to learn diving at club. I think I broke my hip.
Yeah, I
Okay.
slammed my hip on the floor like
I think I
multiple
like
times.
right on my stomach, right in my hip. Like my knee went very low in my stomach.
And now my shoulders hurt, my chest
Like
hurts,
I'm so
my
done.
lats.
Well, I'm hoping this year we'll be doing some driving drills. I think like we're gonna
do that sometime this
get elite this year, team. Shh, you're too loud. Thank you.
Yeah, diving is really hard to learn though. Like we did 30 minutes on it and I'm still
you have to do like pancake drills? You have to do pancake drills for like deadly brutal ones? Yeah, so eventually you'll do like pancake drills, which is for like deadly last ditch ones, where all you can do is get like literally your flat hand under it like that. Yeah. Well, this is why like the serious ones don't even need knee pads, right? Like they know how to hit the deck without tearing their knees up. But that's, elite stuff.
You told me it was a better. You yourself told me.
No, I said just interchangeable. I don't care.
Let's go.
Okay, guys, you should know this on test day. What don't I want at your table? 3 people, books, bags, binders. So come on, let's get this set up the way I like. Where do I want your binders? By the way, where do I want your bags every day? Because this is something that seems in the last 2 weeks to have like gotten brutal. That's right. Come on. So let's be ready. Move. Thank you. You're into your own time.
I just gotta sharpen my pencil.
Absolutely. You take your time. I'm fine with that. Though again, a habit you should start breaking yourself of, because when you're in university, they will not be letting you listen to music while you write tests, or any of the other various things we do to make it more pleasant.
Tests aren't pleasant.
You know what? Can I tell you? Like the counterpoint to that is, I used to always say to myself, for every test I ever wrote, for every exam, I used to say a little prayer and just say like, let me just show what I actually know. And like, let me get the mark I deserve. But I honestly think like, if you just think of it as an opportunity to show what you know, and a challenge to figure out, the tricky stuff. In other words, boy, your attitude is going to determine a lot about both how effective you are, but also how much you enjoy writing a test. And I know it may sound weird to talk about enjoying writing a test, but honestly, like, you know this stuff. Just keep your head up and do it. You do know this stuff. I promise you know this stuff. So, given that you know this stuff, you're going to be at your best if you kind of just chill out, right? Just chill out. It's just test. So let's say, oh my God, it goes horribly wrong and you get all the questions wrong. Like, what then? And the answer is like, I don't know. Then tomorrow's Tuesday and everything's fine, right? Like, it's test. You want to do your best. You want to make a good showing, and like you can only do what you do, and then you move on. Yeah. No. No, do your best. Just do your best. And you'll either do great or you won't. But no. And I don't say that to be mean, but you got this. You got this. Just do your best. Would somebody hand these out to me? Oh, so many hands. But I think I saw Annie's hand first today. I think she was complaining about that sore shoulder, and then she's like, bam, look at this. Bam. I'm gonna stop. Right now.
Enthusiastic.
Right? Okay. Did you grab a calculator? Do you have a calculator? That's important. Do you have something to write with? Do you have an eraser? Do you have a highlighter? I like highlighters. I'd grab some highlighters. God, I love highlighters. Do you want some highlighters? You sure? Are you sure? You have your own? Yeah, you bet you can.
Can I also have lunch today, please?
Yeah.
But not Thursday and Friday.
I was really sick. I slept.
Thank you. Alright, I'm done, Annie.
After school on.
No, I'm joking. I actually know what I'm doing.
Yeah, Scarlett, despite my being mean and unkind, I am glad to see you and that you're doing well. And just also just do your
I'm
best.
not doing well. I haven't eaten a
Oh,
food that is.
no.
I haven't eaten a food other than applesauce since Wednesday.
Yeah,
I hate
listen.
applesauce.
Listen, just do your best, okay? That's all you can do. And don't worry, we'll talk. Who asked for the paper? You asked for the paper. Have you actually not eaten since Wednesday?
I've eaten, but I can't eat solid food yet.
Oh,
so it's just
Scarlett, that's
applesauce.
awful.
You know how gross applesauce is for like the 50th time?
Yes. Yeah,
And my
that sounds
family got
horrible.
chicken
You too.
wings.
It's Friday.
And they just
And you
don't come in
didn't?
the kitchen. I can still smell them.
Oh, I'm sorry. It's okay. But having said that, guys, I want to be, listen. I want to be pin drop quiet in here. All right, we're writing the test, not having social hour.
You got visitors.
What did I just say?
They want the computer card.
Then they can come get the computer card. Make sure you unplug it, please. Yeah? Oh.
Oh, sorry, were you giving us a
That I got. Question, answer. We discussed it in the first 5 minutes of class one day when many people were late. Do your best.
Is that paper to do
Yeah,
extra
that's
work on?
extra paper. If anyone else like extra paper, you can show your hands. You can also just ask me at any time. Okay. All right, bon chance. Do your best.
Are you actually counting that first question?
Of course, it's a question.
What?
Get to work.
What? What?
Sorry?
What a great way to
Put your hand down.
What the hell?
Put your hand down. Okay, good. I actually need to do the attendance, so sit down. I'll come to you.
1 second. How long are the roads exactly?
That's really for you to figure out. Grab a seat.
What?
Guys, skip it and move on.
How long are the roads?
There's no information there.
It's definitely not. Anyway, get to work.